Welcome to today's Ask the Expert. We are excited to welcome Dr. Xavier Gidrol and Emily Tubbs to speak about promises and challenges in pancreatic organoid development. And just a little bit about them. Dr. Gidrol received his PhD in molecular and cell biology from Aix-Marseille University. He performed postdoctoral research at Harvard University School of Public Health and at the Institute of Molecular and Cell Biology, Singapore. He specializes in transcriptional regulation of gene expression and served as Associate Director of R&D Xenometrics, Inc., a spin-off startup from Harvard University, where he was interested in global approaches in genomics. He then joined in 2000 the newly created Functional Genomics Laboratory at CEA in Paris and managed it until 2008. This laboratory focused on large-scale functional genomics approaches to analyze proliferation and differentiation, balance, and oncology. In 2009, he was appointed director of a new laboratory, Biomics, at CEA in Grenoble to use microsystems for cell biology, where he focuses on 3D organoids, organoids on chip, and RNAi-based functional genomics in oncology. He has authored more than 140 publications and has 6 patents. Emily Tubbs obtained her PhD in 2014 from University of Lyon 1 on the importance of intracellular mitochondria-associated ER membranes in type 2 diabetes. She performed her postdoctoral research in the Clinical Research Center of Malmo on identifying new therapeutic targets and drug repositioning for the treatment of diabetes. Then she joined the Laboratory of Fundamental and Applied Bioenergetics in Grenoble, particularly on the European project BioCapan, to develop a bioartificial pancreas based on co-encapsulation of pancreatic islets and MSCs. And she joined Biomics in 2021 as a young researcher on pancreatic islet organoids called Langerhanoids towards personalized medicine for type 1 diabetes. Welcome to both of you. Thank you so much for joining us from Paris. And we're really excited to see what you have to present. We'll take questions in the chat during the talk, and then we'll take them at the end. So thanks again.
Thank you very much, Monica. We do appreciate bonjour tout le monde. That will be my French accent. And I'm sorry for my unbearable French accent. I'll try to get rid of it, but it's not working well. So
don't be sorry. We like it.
Thank you. So we're going to start right away. You keep time on.
Yes, I'll keep time.
Okay. I'd like to start with definition because it's always useful. So we work on organoids, spheroids, tumoroids. There's a lot of names around. I like to define them. So whenever you start with stem cell, whatever they are, induced, embryonic, or adult stem cell, you put them in a low adherence plate. They do form spheroids. So I remind you guys that the suffix oid means which resemble to. So if they do resemble sphere, they aggregate together, desperately want to be together to form a spheroid. Then, depending on the intrinsic properties of the cell or the extrinsic properties of the medium, physical and chemical cue, you will target the cell toward different fate, either organoid. Here we have prostatic organoid that we generate in the lab. You see that they are really differentiated. So they start to look like the organ, so the name organoid, because they resemble to a prostate structure, this grape-like structure, which is differentiated. And with other cell, prostate cell again, they will never ever, like this one on the top, where they will never ever differentiate, still say a spheroid, and they look like more tumor than organ per se, so the name tumoroids. So we do generate all that. We always go through a state of spheroid. Then once again, depending on the chemical cues, physical cues, the extrinsic properties of the environment and the intrinsic properties of the cell will generate this different fate. Also, we are all generating, I mean, there are plenty of labs in the world generating organoids from stem cell, whatever they are. We all face several limitations. Organoids are very promising indeed in basic research, in personalized medicine, in regenerative medicine, in pharmacological industry. There is a lot of potential, but they are facing limitations. The first one is the excessive variability, as you can see here. I hope you see my mouse. Indeed, they auto-organize, as I mentioned here, they self-organized. And as they do self-organize, we do not control their fate completely. And we have a lot of variability in the results, the object we obtain. Furthermore, starting with stem cell, we have a partial maturation. In the end, we have structure, even when they are differentiated, they look like more the embryonic organ than, rather than the adult organs. So that's some limitation of the organoids. So I will illustrate that. If you see this organoid we have generated in the lab is circumvented by this yellow line. There is some green and red at this stage. You don't want to know more. So we are very happy with that. And the cell together, the stem cell, when in this medium, put in this medium, generate something like looks like a painting of Jackson Pollock. I took an American painter, of course. They really look like that. And another day, the same cell in the same medium, they will generate an organoid again with red and green. So we are happy with that. But this time it looked like a painting of a German guy called Edward Munch, which is really well known. So besides the fun illustration like that, I want to illustrate the fact that we do not control the fate of the 3D structure, and that's a limitation. Furthermore, in this review of Roger Cam Lab in 2021, they were mentioning that indeed we lack growth when we grow organoid on hydrogel or even on medium, on low adherence plate. We do have some growth, but there is limitation because indeed there is no vascularization, and at the center of the organoid there is some necrotic arc. So that's another limitation, a growth limitation of the organoid. So we think that with others that having this organoid on organ on chip, which was something completely different, where here on organ on chip we don't tend to look like the organ, resemble the organ, just mimic a function. So in the end, you have something which look like more like a domino, not like an organ, but mimicking well a function. And we thought that having organoids on organ on chip or on chip, organoids on chip, we will succeed in limiting or solving, at least partially, some of the limitation I was mentioning just before. So we've been doing that. We are generating chips in the lab. These kind of chips, they are the size of a credit card, actually, with a serpentine structure like that. A restriction here where we can trap an organoid. So we're going to generate the organoid outside of the chip using classical way to do that, classical protocol. Then we do trap the organoid on the chip automatically, as you can see here, here, and here, slowly here. Just by withdrawing with one syringe through this hole in, and withdrawing here, you can see the picture here, we can trap automatically in this restriction, micro 3D restriction, different 3D objects, beads, organoids, spheroids, tumoroids, and it's working perfectly. It's you can parallel, make it parallel like here, or even in serial, having 10 trap like that, 10 restriction. So it's really useful and very efficient to trap organoid onto that. So we've been, I will remind you quickly the way it's working. So we have a mixture of organoids, an hydrogel, which is not reticulate yet, and cell which are endothelial cell, UVEC most of the time, that we're going to use as a gate to enter the organoids. I'll come back to that. Then we just we withdraw air into it, draw air into it, and then the growth medium without changing the syringe one after the other. And we're going to trap the organoid spheroid into the restriction. It's surrounded by the gel, which is going to start to reticulate. We send air into that to push the extra amount of gel. And you can see that there is a residual part of the gel remaining in the microfluidic tube, as well as in the restriction. And then we send the medium onto that, and we culture grow and grow on for a while, up to a month if we want, with a constant flow of culture medium into that. So to show you otherwise, you can see that the microfluidic channel is square like that. We're making a square microfluidic channel. And as I mentioned before, and as you can see here, you have some residual gel, a mixture of gel and endothelial cell, UVEC in here, which remain into the microfluidic channel. And you can see it with this draw here on the side here. And in reality, it's like that. This is a real image where we have had some fluorescence in the gel. So you see the gel remaining in the microfluidic channel. You see the gel in the restriction, the trap, the microfluidic trap. You see the spheroid here, or the organoid, whatever. And then we grow that. So how does it look like? It looks like that. So here we have a trap, what we call mesenchymal spheroids. It's a mixture of GFP UVEC, GFP labeled UVEC, endothelial cell band, and fibroblast. And as you can see here, we have the GFP UVEC into the spheroid here, which is green. And in the channel, we have this gel I mentioned before with RFP label, red label UVEC that you can see here. And when we grow that with a culture medium into it, oops.
Maybe go back.
Go back to.
I think you can try escape.
Escape.
Oh, it froze. We're sorry. Maybe we can switch.
Sometimes you can reload.
Oh, good.
Here we go. Let's try again.
Just have a little break.
Here we go. I hope you still see the screen, Monica? And
Yes.
everyone? Okay, great. So after at T0, we see this RFP UVEC. They start to grow and make a network, as you can see here. Similarly, the spheroid, the endothelial cell within the spheroid will grow up, and eventually they will connect, and they will anastomose. So we are already addressing one of the limitations I was mentioning at the beginning regarding organoids, this high variability, excessive variability. Here, because we control the size, we control the place where they are in the chip, we control the growth of the medium and the network, endothelial network surrounding it. We limit variability, and besides that, as you can see here on this close-up, we have a real anastomosis with red cell, red UVEC connecting to green UVEC. And it was what we wanted in order one day to perfuse the organoid from the external, like this drawn here. So to show that, we've shown that we have anastomosis. We want to see that, wow. Beads are entering the gel. So you can see here fluorescent bead entering the network, UVEC network, going into the chip and really perfusing the organoid in the heart of the chip. So we tried that with blood vessel organoid. We generate blood vessel organoid from IPS. I won't go into the detail, just go faster into it. We checked that we do not have just an endothelial network like before with UVEC, a real vascular network with all the neural cells. And you see that capillary with all lumens in here. So we put that, wrap that onto a chip exactly the same way. And as you can see with a movie here, from D0 to D13, we have a vascular network this time coming and growing up out from the blood vessel organoid we have been generating from IPS and into the gel onto the microfluidic channel. Then we wanted to see whether or not we had, I go too fast, we had beads coming in, and you can see these red fluorescent beads going to the channel, showing that there is indeed a perfusion. And if we overlap of the traces, the fluorescent traces, you can see the road that the bead takes inside the network. But we're not sure at this stage that we are perfusing per se the organoid, whether or not it's just surrounding the organoid in the network around it, but not into it. So we wanted to address that further. And indeed, we show that by using antibodies against CD31, a marker of endothelial cell, which is labeled in red here, we brought it through the chip into the organoid. And because the endothelial network is labeled in green, you can see here that there is some part on the chip where there is red, but no green. So that's the interesting part to us, because it shows that the antibodies we brought through the chip, through syringe and the chip, enter the UVEC network. And as I mentioned before, it's a gate to the organoid, and then enters the organoid, and you can see this red part without green. It's even better here. You can see the red organoids, which is labeled by these antibodies against CD31, and not green at all. And you can see some markers that showing that it's a vascular network indeed, and with hollow structure as well. So we went further onto that also by showing that beads that will enter through the microfluidic chip will go into the organoid to really show that we can perfuse the organoid. And then can see here the fluorescent bead into the BVO. Then we go here and we're gonna close up or close in here in region where there is a red label showing that we are inside the organoid, and this blue dot which are big. And a bigger close-up of this region here, you can see here, and there is plenty of fluorescent bead inside the endothelial network, even vascular network. And with confocal analysis, you can see even better here. With this red part, we want to see bead in the red part, that means inside the heart of the organoid. And you can see it with this cut here, where by reconstruction, we can see one bead in yellow here, really within a network, a tube, a lumen of vascular network. So we were very happy with that. And we show that indeed we have reduced variability, but besides that, we achieved perfusion, which what we wanted to achieve. So to the end, I say that one other limitation of the using organoid, iPS generated organoids is a lack of maturation. They resemble more the embryonic organ rather than the adult human organs. So to show that, we've been comparing organoid vascularized on chip, the way I showed you just before, with biophysical properties or conditions which resemble the flow inside the body, and compare that with an organoid transplanted in mouse, which is a way to mature the organoid and make them resemble more the adult organs. So we compare that by comparing transcriptome, and as you can see, the color here, yellow and green, the two transcriptome that resemble more to each other are the one of the transplanted organoid into a mouse, and the one with the vascularization and perfusion with flow on chip. So that means maybe nothing to you, but to me it's very important, because it's, I'd say for the first time, that we can maturate an organoid almost as efficiently as in a mouse by just vascularize them onto a chip with a flow. The flow is really necessary. You can see here, for example, there was no flow, but it was not maturing as well. So we've been trying, it's very versatile technology. I will go pass the mic to Emily. We've been trying this mesenchymal spheroid, as I mentioned to you, the iPS-derived blood vessel organoid. And then we have been playing on islet, and the reason we are talking here the sugar science, sorry, it's because we work also on islet, and that Emily doing that, and go give her the mic. We switch.
it's almost the Olympics, so we're ready for the passage.
That's great.
Thank you very much, Monica, for the introduction, and thank you, Xavier. It's really a pleasure to be speaking today to the Sugar Science. Thank you very much for having us. Like Xavier mentioned, we are particularly interested in many different systems, but our research also focuses on islet spheroids, and this is how we use this chip that Xavier previously mentioned. You all know that islet transplantation is indicated for patients who have unstable type 1 diabetes, or for patients who have already had kidney transplant. You're very aware crowds, I won't go into too many details. Just a short reminder that from a donor, we can isolate, we collect the pancreas and isolate islets, and that these islets are then transplanted into the portal vein, into the liver of a recipient patient. And then cells are nicely placed in the liver, and results are really encouraging so far. In this study from 2021, we can see the glucose traces, the average blood sugar levels during 24 hours for one patient. So that was before transplantation. We can see that the patient was out of the normal glycemia in this yellow area here, and that 1 month after transplantation, this same patient had a restored glycemia. So results are extremely promising. However, some limitations remain. There's a big lack of donors, as you know. Recipient patients need immunosuppressive treatments, and that in the long term can lead to infections, cancers, also alterations of the renal function, and also impact actually the graft. Also, one of the major limitations is the loss of graft effectiveness that's observed usually around 5 years, causing early inflammation, acute chronic hypoxia, rejection, and in some cases, recurrence of type 1 diabetes. We strongly believe in this chip that Xavier presented previously with the very encouraging results we had. And this is what I would like to describe into more detail with you today. What we did is the organoid, which is quite a mouthful to pronounce. But what we did is take endocyte beta H5 cells here, so insulin secreting cells that we can get from human cell design, and fibroblasts, and as well as endothelial cells, which are labeled RFP. We put them together in a well. They nicely formed a pre-vascularized islet organoid, and then we put them on chip. On chip, we put a microenvironment around, which is composed of UVICs, which are this time GFP. Like Xavier mentioned, when we have these red and yellow and green cells, what we hope is to see connections between these cells. Then we performed a classic functionality test, GSIS, for the glucose stimulation and insulin secretion assay. So on the chip, we can do that. We have the entry and the exit, and we can do the incubations like we would do in normal bench studies. Here we have the insulin concentrations for the different conditions mentioned here, either in well, static without vascularization, static with vascularization, and then the impact of flow. What's really interesting is if we have a focus here on the stimulation index, if we have only the environment, so only the vascularization or only the flow, we don't really see an impact on the few on the experiments we did, but it was really quite noticeable that both flow and vascularization, both together, had a positive impact on the cell functionality over time. So we were quite happy to be able to show this. We didn't really know what to expect, but it shows that on chip we can improve functionality of cells. But what we didn't manage to do in this study so far is that we didn't manage to do this nice perfusion that Xavier showed with beads going through, because I mean, this network is nice, but it should possibly be improved to have this perfusion. So for this purpose, we have launched a PhD project, which has this idea behind of having a pre-vascularized organoid and a vascularized supporting gel, both having on chip and hopefully getting this nice vascularized organoid on chip. on this serpentine design that Xavier explained. And this is a work currently done by an excellent PhD student that we have in our group called Melanie Lopez. So hopefully in a few years, she'll be able to present, if you agree, to the sugar science, and hopefully have really exciting results to show you. What we can tell you so far, here's a scheme of the insulin-producing cells. I'm sorry it's in French, but we thought we would French it up a little bit. It's in a French paper that we show that these insulin-producing cells can be obtained by a variety of ways. You all very well know, I'm sure, using either IPS or also from Langerhans islets, or these beta cells I mentioned from endothelin H5, or transdifferentiated by other ways. What we usually do in the lab is to do these pre-vascularized organoid. We use these insulin-producing cells. So in our lab, we use IPS, human islets, and beta cells from endocytes. We have them single cells. We add endothelial cells to them and fibroblasts, all single cells, put them together in a well, ultra-low attachment well, of course, and then they auto-organize. And we really have this focus of letting them auto-organize. And what it looks like is, hopefully it will work, is like this. So this is just a video tracking of the cells, and they have this need to get together and to adhere. And this is how, in the end, after approximately 6 to 7 days, we obtain these nice-looking 3D structures that we call organoids. To pre-vascularize these cells, we have different alternatives. So the one I just mentioned, starting from these organoids and insulin-producing cells, adding endothelial cells and fibroblasts, and we obtain this 3D structure. But we also have another strategy ongoing in our group, which is adding blood vessel organoids. So the same structures as Xavier explained previously, the blood vessel organoids, which have this complex vascularization structure, and putting them both in culture together. So this is what I'm going to explain briefly to you. What we do is we put an islet and a BVO, so these two 3D structures together in one well in co-culture, and wait for them to meet if they decide to auto-organize or not. And what we see is self-assembly of these two structures. Here, this is also what you can see by taking pictures at several time points, at day 1, day 3, day 5, or 14. This is for just an islet alone to see as a control. It's becoming over time, a BVO alone. And here we have BVO and pancreatic islets in co-culture for 2 different donors. And here the magnification is further, but here we can zoom in and see that with time, we have this engulfment, this meeting, this merge of these 2 structures. And this is the same for the second donor that we performed. We can see this connection between the 2. We wanted to go a bit further and check the becoming, and so we did labeling for glucagon and CD31. On the left here, you can see BVO alone. Here you can see BVO and islet in co-culture at day 3, and here at day 7. Unfortunately, we can't have the same fusion, so these 2 are not the same, but we can still see that over time at day 7 here, I can play it again. We have this connection between the 2. And here is a screenshot and maximum projection for these cells. And here we can see they meet. And here what's really exciting is at day 7, we can see that this connection really happens. This could possibly be taken on for further, and hopefully maybe we would see a further merge of these 2 structures, but so far this is really encouraging. And what we looked at also is the functionality, because of course it's nice for them to meet and to vascularize for applications in islet transplantation, but we also want them to be functional, and that's the main element that we looked at. So here's the insulin secretion for the first islet donor, the same as you saw previously, here for the second donor, and underneath the stimulation index. And what's really interesting is at day 14, we can see that in light gray, here's the islet alone. With time, we have, of course, this loss of functionality. But what's really interesting is that when we have both BVOs and islets in co-culture, we have a significant increase. So it means we have maintained the functionality of the eyelids thanks to the presence of BVOs. And this was confirmed with the second eyelid donor. So of course, this is rather preliminary work. We wanted to share it with you. It's only a preprint for now in bioRxiv, but we are currently working at increasing these observations and hopefully taking it a step further on tube. So for this, we'd like to thank our group in Biomics, also our colleagues at the CEA Liti in Fabrice Navarro's group, and all our different partners and collaborators, especially diabetologists and the eyelid isolation centers, who are really helping us in obtaining these data. And thank you so much for hosting us today.
Wow, beautiful. I mean, some beautiful work, and you've accomplished, it seems, you've accomplished so much in such a short period of time. I want to open up, to questions. If people can drop some questions in the chat, we'd be happy to answer them.
See you also.
I also, I wanted to ask you guys, with this sort of the co-localization or the vascularization and the beta cell developing together, is there any kind of timing? Is there a time constraint for those to, merge most robustly, or, create the best situation for them? Is there a certain time window for it to?
Yeah,
to
I
really
think
work?
for now we're trying to see what happens just by letting them be. Maybe we could influence the speed of vascularization, but we haven't really tried this. For now, we're just letting them auto-organize. But of course, this chip has a tremendous potential, and we can imagine drug testing and seeing if some drugs would accelerate this network development, and that would be really interesting.
Mm-hmm.
What seems to be very important is the flux, actually. Our breakthrough, I guess, is in that field, because for the first time, to the best of my knowledge, we have some flux which mimics the physiological one in terms of shear stress, in terms of velocity of the blood. It's not blood here, it's a gross medium, but and we figured that it was very important indeed. So for the BVO and eyelid fusion, so to date, we are just at the stage without flux, and we think that we can improve that both in terms of functionality, but in terms of duration with the flux. And that into two directions, I would say, two applications. The first one is for graft and eyelid graft. We think that having BVO fusion first might improve the grafting and the vascularization of the graft, including maybe reducing the immune response, but that we don't know yet. Having, for example, BVO from the patient and the eyelid from a donor, a mixture of allograft and autograft, if you want. And the second application was more on the part I was mentioning at the beginning, is having eyelid onto a chip under flux, so we can come with a T cell, for example, or immune cell, which will perfuse into the eyelid. And that might be very interesting to better understand the etiology of the disease in terms of autoimmunity, but also to screen for immunosuppressor and so on. So the two aspects are to keep in mind.
Yeah, no, if you could emulate the very first, the beginning of pathology, that would be really exciting. Here's a question from Jim Wells. Sorry, I missed how the vascular organoids were generated. What were the starting cell types?
It's an iPS-generated blood vessel organoid. We've done that with Joseph Penninger's lab. Joseph Penninger is both in IMB in Vienna and in British Columbia, Vancouver, British Columbia University. So they start with iPS and we generate them. We have a slide, but from the iPS. So they do a really vascular network. It was the reason I was insisting between we're comparing what is an endothelial network only with UVAC from a vascular network. Thank you, Emily. You can see on this slide that we have pericyte, we have tight junction, we have adherent junction, we have smooth muscle cell as well. It's not on the slide, but we labeled them with SM22. So we have all the mural cell as well as the basal layer. So they are PS-derived blood vessel organoid, but they were not really mature, and unless you put them, you graft them in mouse. And we've shown here that vascularize them onto a chip is almost equivalent to maturation in mouse. So that's also promising.
Okay, great. Thanks for answering that. And elaborating on that. That was great. Question from Ying Dang. Thank you for this nice presentation. In your islet plus BVO co-culture, what media do you use to maintain both organoids? Merci.
Well, we use a mixture of both. We thought both need their specific medium, and our choice was made to pay half and half for each. And this, of course, was done also on the control.
Okay, yeah, that's great. And this bioarchives paper and the other papers you mentioned, I did drop into the chat, so those are available in terms of details and protocols.
Thank you.
Yeah, for sure. This is just exciting. It's very exciting times. You guys have kind of come in sideways, from other interdisciplinary, field, I guess, and really come up with something very interesting. And it will be, I think it will be of interest to many people in the field of type 1 diabetes. So congratulations. Oh, here's a question from Peter Wang, I think. Yes, asking to unmute. Go ahead.
Thank you, Monica. Great talk. I also have a question for the co-culture of these two type of organoids. My question is when you co-culture these two organoids, looks like there's a vascular structure connected. My question is for these endothelial cell structures, is migrate from the vasculature organoids to the islet organoids, or the cells differentiate in the different organoids and they merge connected?
So far, but it's only been to day 7, so we think they still keep their individuality, if you want, because the labeling that we did here for CD31 and glucagon seems rather specific, but it's a possibility considering IPS, I don't know if you agree, but that they could become something else, of course, going further into the identity if some cells are changing and differentiating, could be a possibility.
I think it's a very interesting question indeed, and we don't know the answer. It's the reason why it's interesting, and we have to learn a lot on that. It's a very the beginning for us. We plan to make a single cell RNA-seq and do that, of course, to see whether or not we transdifferentiate into a new cell, which cell population we have into that, having the BVO alone or the islet alone, and the two together. We will have more answer on that in a few months. You have to come back to us on that. At the moment, we do see mainly the association of two, but without really interpenetration and transdifferentiation. This has to be addressed here.
Thank you.
You're welcome.
Yeah, that's great. And this is, you've gone up to seven days, right? That's your...
Yes.
It's already...
Yeah, so it'll be very interesting to see how far you can go out and what morphology develops. It's really exciting.
Yeah.
Well, in the interest of time, I just said we'll say thank you so very much, and I'm certain that you'll get a lot of watches once we release this recording onto our website, and I encourage people to look for it. It's really interesting, and thanks again for taking the time to present.
Thank you, Monica.
Thank you.
Thank you.
Yeah.
We're happy to
Yeah.
do that with you guys. Thanks a
Thank you.
lot.
It's mutual. Thanks again. Have a great rest of the day. Bye-bye.
Thank you. You
You
too. Bye.
too.